Welcome to the AraENCODE database. In the AraENCODE database, we have integrated Arabidopsis epigenomic datasets such as ChIP-seq, ATAC-seq, MNASE-seq, and BS-seq, 3D genome datasets including Hi-C, Hi-ChIP, and Chia-PET, as well as transcriptome datasets like RNA-seq and ncRNA-seq. At the top, you'll find a navigation bar that allows you to access different modules. Now we are on the home page. Here, you can see an illustration of chromatin's three-dimensional structure. The text on this illustration serves as a navigation menu, and you can click on them to navigate to the corresponding module pages. On the home page, you can use the quick search feature. For example, if you enter the PHYB gene and click on Search Gene, you will quickly retrieve information about this gene. In the WashU browser, you can view various distribution maps such as RNA-seq, histone ChIP-seq, and DNA methylation at the PHYB gene locus. Here, you will find information about chromatin loops, histone modification, chromatin accessibility, gene expression, and DNA methylation related to the PHYB gene locus. You can access the WashU browser page by clicking on WashU Epigenome Browser in the Browser section of the top navigation bar. In AraENCODE's WashU browser, the default reference genome is TAIR10 for Arabidopsis. You can import data into the WashU browser by clicking the Tracks button. Public data hubs contain published epigenomic and 3D data related to Arabidopsis that have been collected by AraENCODE. Of course, you can also add tracks through other means. You can select the data type you are interested in and click Assay to expand the selected data type. By clicking on elements in the matrix, you can further select specific data. Clicking the plus icon on the right will import the selected data into the WashU browser. In the WashU browser, you can use the zoom bar to scale the current chromosomal region. By clicking on the corresponding track, you can customize its display format. To access the HiGlass browser, you can click on HiGlass Multiscale Browser in the Browser section of the top navigation bar. Similarly, the HiGlass browser also defaults to the Arabidopsis TAIR10 reference genome. You can click on the Launch HiGlass in Full Screen button in the top left corner to enhance your viewing experience. In HiGlass, if you want to delete a track, simply click on the X button in the top right corner of that track. To add a track, click on the plus button in the top right corner of the track. For example, if you want to add a track in the center, select the data of interest to view its heat map in HiGlass. You can also add a compartment track at the top and choose the desired display format. AraENCODE also supports viewing loops. Similarly, add a loop track in the center, and then you need to drag and zoom in the heat map to locate specific loops. Each box represents a loop. Of course, you can also enter the chromosome region you want to search for in the location bar at the top. For more detailed instructions on how to use the WashU and HiGlass browsers, you can refer to the tutorials provided on their official websites. You can access the histone modification search page by clicking on Histone Modification in the Search section of the top navigation bar. Within these drop-down menus, you can select different tissues and histone modifications in Arabidopsis. In the two sections below, you can choose the gene or chromosomal region you are interested in. For example, enter PHYB in the Gene ID input box and click the Search Gene button to obtain the results. The table displays the ChIP-seq results of H3K4Me3 in all seedling tissues of Arabidopsis in AraENCODE. By clicking on the ID column, you can navigate to the SRA database or GEO database. Alternatively, clicking on the histone column will open the WashU browser window below, automatically positioning it to the chromosomal region you entered, showing the ChIP-seq signal values of H3K4Me3. This graph illustrates the distribution percentage of H3K4Me3 signals on different gene elements. Lastly, this graph displays the distribution of H3K4Me3 signals within 2 kilobytes upstream and downstream of genes with varying expression levels. You can access the chromatin loops search page by clicking on 3D genome in the top navigation bar of the search. Here, you will also find the HiGlass browser, which can be loaded by a single click to open the HiGlass multiscale browser. In the two drop-down menus below, you can select different tissues and libraries to search for chromatin loops. You can also choose the minimum PET count on the right side. Similarly, in the two sections below, you can select the gene or chromosomal region of interest. For example, input PHYB gene in the Gene ID input box and click the Search Gene button to obtain results. The datasets involved table contains all the data related to the gene you selected based on the chosen tissue and library. The results table provides specific information about each interaction of PHYB gene within the loops. Clicking on the ID column allows you to navigate to the SRA or GEO database, while clicking on the PET count column automatically opens a WashU browser window below, displaying the imported data. At the bottom, there is an interaction network where you can click on individual circles to view specific gene elements involved in the interactions. You can also drag the elements for easier exploration of the interaction details. If you only want to view the interaction information of a specific dataset, you can enter the corresponding dataset ID in the Legend window. You can access the Open Chromatin Region search page by clicking on Open Chromatin in the search section of the top navigation bar. In the three drop-down menus, you can select different tissues and libraries to search for open chromatin regions. Similarly, in the two sections below, you can select the gene or chromosomal region of interest. For example, input PHYB gene in the Gene ID input box and click the Search Gene button to obtain results. The datasets involved table contains all the data related to the gene you selected based on the chosen tissue and library. The results table provides information about the chromatin accessibility of the loci where the PHYB gene is located. Clicking on the ID column allows you to navigate to the SRA or GEO database, while clicking on the histone column automatically opens a WashU browser window below, displaying the imported data. You can access the chromatin state search page by clicking on chromatin state in the search section of the top navigation bar. After selecting the desired tissue, you can choose the gene or chromosomal region of interest in the two sections below. For example, input PHYB gene in the Gene ID input box and click the Search Gene button to obtain results. The table represents the chromatin state on your input gene region in 200 BP intervals. These three images display the chromatin states on the search regions in different formats. At the bottom, there is also a WashU browser window that showcases the epigenome landscapes of the gene region you selected. You can access the DNA methylation search page by clicking on DNA methylation in the search section of the top navigation bar. In the two sections below, select the gene or chromosomal region of interest. For example, input PHYB gene in the Gene ID input box and click the Search Gene button to obtain results. The DNA methylation level in gene body table displays the DNA methylation levels on the body of the selected gene. The DNA methylation level in gene promoter table displays the DNA methylation levels on the promoter of the selected gene. Clicking on the GEO column will take you to the GEO database. Clicking on the source column will open a WashU browser window to showcase the methylation levels of that data. This bar chart presents the DNA methylation levels at different samples. In the legend on the right, you can select different methylation patterns for the gene body or gene promoter, with colors indicating display and gray indicating no display. The slider below allows you to filter different samples. You can access the transcriptome search page by clicking on transcriptome in the search section of the top navigation bar. In the Gene ID input field, you can enter the gene ID to obtain gene expression information for that gene. Similarly, you can enter the miRNA ID in the miRNA ID input box to obtain miRNA expression information. The results are displayed in the form of box plots. By clicking on Statistics in the Data section of the top navigation bar, you can view all the datasets collected by AraENCODE, displayed in the Datasets table. The line graph in library types and years shows the distribution of libraries in different years for the data collected in the AraENCODE database. The Venn diagram represents the specific data distribution for a particular year, which changes as you move the mouse on the line graph. The bubble chart in Tissues and Variants displays the proportion of mutant and wild-type data in different tissues in the AraENCODE database. The bubble chart in Tissues and Libraries shows the proportion of different libraries in different tissues in the AraENCODE database. You can access the Quality Search page by clicking on Quality Control in the Data section of the top navigation bar. Here, you can enter the dataset's ID of interest in the appropriate input fields based on different libraries to view the quality of the data. You can download the data from the AraENCODE database by clicking on Download in the top navigation bar. For example, clicking on 3D Genome allows you to download compartment data, loop data, or Hi-C matrix information that you are interested in. You can check relevant information about the AraENCODE database by clicking on News in the Help section of the top navigation bar. It provides a brief record of the establishment and improvement process of AraENCODE, and any new features added to AraENCODE will also be updated here. Clicking on Pipeline in the Help section allows you to view the workflows for different libraries in the AraENCODE database. If you want to get in touch with us, you can find our contact information in the Contact section of the Help menu. If you wish to contribute your interested datasets to the AraENCODE database, you can submit your datasets through the Submission option in the Help section.